So picture this scene, because I guarantee it has happened to you. You just stepped out of a hot shower. The bathroom is totally steamed up. You're reaching for your towel, and you accidentally brush your bare hand against the metal doorknob.
Oh, yeah. And instantly your nervous system tells you it is freezing cold.
Right. But then you touch the wooden door frame right next to it, and the wood feels totally fine, like just normal ambient room temperature.
Even though they're in the exact same room.
Exactly. Logically, as a functioning adult, you know the metal doorknob and the wooden door frame have been sitting in the exact same room for years. They are fundamentally the exact same temperature.
Right, but your brain is screaming that the metal is practically made of solid ice.
It's wild.
Yeah.
And it gets even weirder than the doorknob, because when you're standing in that shower, feeling the water wash over your skin, you are actually experiencing a brilliant biological illusion.
You really are. It's incredible.
Because mammals do not structurally possess receptors for feeling wetness, when you touch a pool of water, your body is mathematically tricking you into feeling a sensation that just doesn't physically exist anywhere in your peripheral nervous system.
We are essentially hallucinating the feeling of water. I mean, it is a phenomenal piece of neural engineering that happens so flawlessly we never even question it.
Welcome to the Deep Dive Lab. Today we are taking a deep dive into a monumental 2025 review article published in Nature Reviews Neuroscience.
Yeah, it was authored by Dr. Mario Carta from the University de Bordeaux, Dr. Mikkel Vestergaard from the University of Copenhagen, and Dr. James F.A. Poulet from the MDC Berlin.
And our mission for this deep dive, we want to figure out how our brain is essentially hallucinating our physical reality. Like, if we don't have wetness receptors, and if metal and wood are the exact same temperature, how exactly does our body encode and invent the perception of temperature?
It's a great question. And for over a century, the scientific community was genuinely baffled by those very things. But we were finally mapping out this invisible sensory circuitry. And honestly, the mechanisms these researchers synthesized are completely upending everything we thought we knew about human perception.
So, okay, I think most of us grew up with a pretty rudimentary idea of how temperature works. I always pictured it like a digital meat thermometer.
Right.
You poke the turkey, the thermometer reads the heat, and a little number pops up.
Yeah, and that was actually a dominant paradigm for a very long time. We called it the labeled line theory.
Labeled line.
Yeah, so back in the late 19th century, researchers started mapping the human skin. They took tiny, precisely calibrated thermal probes and just touched different spots on human subjects.
Okay.
And they noticed that some microscopic spots on the skin only reacted to cold probes, and some only reacted to warm probes.
So they drew the most obvious conclusion, right? Dedicated wires.
Exactly. Dedicated, independent wires. The logic was simple. You touch a hot stove, the hot wire fires, sending a straightforward electrical impulse straight up your spinal cord, right into your somatosensory cortex, and your brain just registers warmth.
Like a binary relay system?
Totally binary. Almost mechanical.
But that basic meat thermometer model has to be completely broken, right? Because if I have dedicated hot and cold wires that just report absolute temperature, a 70-degree doorknob shouldn't feel colder than a 70-degree piece of wood.
Right. The wires would just dutifully report 70 degrees for both.
Exactly.
And that discrepancy is exactly what began tearing the labeled line theory apart. I mean, human perceptual quirks just did not match a simple wire model.
Like what kind of quirks?
Well, the mathematical output of our nervous system was completely nonlinear. Take reaction times, for instance. If you measure the neurological reaction time of a human detecting a cool stimulus, it takes about 0.5 seconds for the brain to process it.
Okay, half a second.
But to detect a warm stimulus of the exact same intensity, it takes about 0.7 seconds.
Wait, if they are parallel wires in the same system, why would the warm wire be functionally slower than the cool wire? They're traveling the exact same physical distance up the arm.
Exactly. It makes no sense under the old model. And furthermore, scientists found we are astonishingly bad at detecting slow, gradual temperature changes.
Oh, really?
Yeah. If ambient temperature drops at a rate of, say, 1 degree Celsius per second, humans barely notice it until the absolute temperature is drastically different. But if it drops at 10 degrees per second, we perceive it instantaneously.
Oh, wow. So our bodies were acting less like thermometers and more like velocity sensors.
Velocity sensors.
Yeah.
Precisely.
Which brings us back to the doorknob. The metal feels colder because metal is highly conductive. It's sucking the thermal energy out of our hand at a much faster rate than the wood is.
Right.
We aren't feeling temperature, we're feeling the rate of heat transfer.
And we are feeling it from the inside out, which is wild. The researchers in this Nature review highlight a crucial biological concept called thermal inertia.
Thermal inertia.
Yeah, we casually assume we feel the temperature right at the outermost epidermal surface of our skin, but we don't. The nerve endings actually responsible for thermal detection are buried roughly 100 micrometers deep within the tissue.
Oh. So when I touch the highly conductive metal, the surface of my skin drops in temperature instantly. But those nerves, buried 100 micrometers deep, are still sitting at a cozy 98.6 degrees.
Yes. Your brain isn't calculating the objective surface temperature at all. It is running a differential equation,
A differential equation.
basically. It calculates the massive sudden thermal gradient, the difference between the rapidly cooling surface and the warm, deep tissue. A fast temperature drop creates a steep gradient across those 100 micrometers, and that triggers a massive alarm bell in the nervous system.
And the wood, being an insulator, draws heat so slowly that the deep tissue has time to cool down synchronously with the surface.
Exactly. It minimizes that internal gradient.
Okay, so if the basic thermometer model was a total bust, how did scientists figure this out? Like, you can't exactly sit a laboratory mouse down and ask it to rate a breeze on a scale of 1 to 10.
No,
Yeah.
definitely not. They had to somehow hack the sensory system to literally watch the invisible math happen. And that was the genius of the methodology analyzed by Carta, Vestergaard, and Poulet.
What did they do?
To map this out, researchers turned to precision high-tech equipment called Peltier devices.
Oh, Peltier plates. Those are thermoelectric coolers, right?
Yeah.
You run an electrical current through them, and depending on the direction of the current, one side absorbs heat while the other side releases it.
Right, and they can shift temperatures almost instantaneously with sub-degree precision. So researchers attached these tiny Peltier plates to the front paws of mice,
Okay.
and they trained the mice to become little thermal reporters. The mice would lick a water spout to indicate whenever they consciously perceived the plate warming up or cooling down.
And while the mice are doing this behavioral recording, the researchers have them hooked up to advanced neuroimaging to watch the specific neurons fire in real time.
You got it. And the neural imaging revealed dynamics that completely rewrote the textbook.
What did they see?
First, it confirmed the reaction time quirk you and I just discussed. Cooling is fundamentally dominant in the mammalian nervous system. Mice react to a cool stimulus in about 0.3 seconds, but take 0.8 seconds to react to warmth.
Wow, so we are heavily biologically biased toward the cold.
Heavily, and the imaging showed why. When a cool signal hits, it triggers a sharp, rapid, high-frequency spike in the neurons. It is a biological starting pistol.
And the warm signals?
They present as slow, sustained, gradual depolarizations. It takes significantly longer for the warm signal to build up enough electrical momentum for the brain to consciously register it.
That's so strange. So where is all this firing actually taking place? Because for the longest time, the concept of a thermal cortex, like a specific address in the brain for temperature, was entirely hypothetical.
Well, they finally mapped it. They found that the primary somatosensory cortex, which neuroscientists call S1.
That's the region handling basic tactile touch and pressure.
Exactly. S1 deals heavily with these sharp, cool signals. However, the conscious perception of both warm and cool, the actual synthesis where you realize, oh, I'm feeling a temperature, that lives in a deeper, more complex hub called the posterior insular cortex.
The PIC.
The PIC.
Yeah.
And this is where the Nature review drops a massive twist.
Okay, here we go.
The researchers started looking at the specific biological ion channels that act as the frontline detectors for these temperatures. Now, there's a very famous receptor in our nerve endings called TRPM8. Anyone who studies basic biology knows TRPM8 is the universal cool detector.
Right. It is the reason why popping a menthol mint makes your mouth feel freezing cold.
Yes. The menthol molecule chemically binds to and hacks the TRPM8 channel, tricking it into firing a cold signal to the PIC, even though the physical temperature of your mouth hasn't changed at all.
So TRPM8 is the undisputed biological cold sensor.
It is. But when geneticists engineered mice to be completely missing the TRPM8 channel, something bizarre happened. You'd expect those mice to be immune to feeling cold, which they were. But those same mice lacking a cold sensor also entirely failed to properly perceive warmth.
Wait, what? Which is a staggering violation of the old labeled line theory.
It totally shatters it.
Wait, I want to make sure I'm wrapping my head around the physics here. A lack of cold sensors equals an inability to detect heat. How does a cold wire biologically gatekeep a warm signal?
Well, it requires completely discarding the idea of separate independent wires. The thermal system actually operates as a disinhibitory gate.
A disinhibitory gate. Okay, let's see if I can translate this. Is it like a traffic light? Like the cold sensor is a red light and the warm sensor is the green light.
Not quite, because a traffic light implies two separate bulbs taking turns. It's more intertwined than that.
Okay.
Let's use a mechanical analogy. Imagine you are driving a car. You have your foot resting heavily on the brake pedal. That brake pedal represents the cool channel, TRPM8. Even at a totally comfortable ambient room temperature, your TRPM8 channels are firing tonically. They are keeping a constant, slight neurological pressure on the brake.
So my baseline state isn't zero. My baseline state is a low-level hum of cold signals.
Yes. Now, say you want to drive forward smoothly. That represents feeling a warm stimulus. To go forward, you don't just slam your other foot on the gas pedal, the warm receptors.
Right. If you hit the gas while still holding the brake, the car just lurches and strains. The signals conflict.
Exactly. To feel warmth properly, the first biological step is to take your foot off the brake. You must actively suppress or inhibit the TRPM8 cool channel from firing.
Oh, I see. The two signals are structurally tangled. The conscious perception of warmth is actually heavily reliant on the sudden silence of the cold receptors.
That is the disinhibition. The suppression of the cold signal biochemically opens the neural gate, allowing the warm signal to pass unimpeded up to the posterior insular cortex.
That is wild. So when I wrap my hands around a hot mug of coffee, my peripheral nerves aren't just shouting, hot, hot. They are simultaneously shouting, stop feeling cold.
Yes.
And that specific ratio of silence from the cold receptors is what lets the brain render the feeling of warmth.
And what proves the sheer brilliance of this design is that we see it preserved across millions of years of evolution. This complex, tangled thermal encoding isn't just a mammalian quirk.
Right, the review mentioned this is highly conserved even in insects, which blew my mind. Like fruit flies, Drosophila are ectothermic. They are entirely cold-blooded.
Right, they do not regulate a core body temperature. They are simply whatever temperature the room is.
Yet, if you examine the neural circuitry in their antennae, which are their primary thermal sensors, they utilize a remarkably homologous setup.
Yeah, they possess specific hot cells and cold cells that interact through this exact same architecture of cross-inhibition.
So evolution found this gas and brake pedal mechanism so computationally efficient at navigating complex thermal gradients that it locked it in early on and scaled it all the way up to the human brain.
It saves the nervous system from having to build two entirely separate, massive parallel processing networks. It's just an elegant...
Okay, so because these two distinct data streams, temperature and pressure, arrive at the exact same neurological address simultaneously, the neurons integrate them.
Yes. When your skin detects a specific drop in temperature caused by evaporative cooling, and that signal perfectly overlaps with the tactile pressure of a viscous liquid spreading across the surface area.
The S1 cortex runs a complex algorithmic calculation and invents the feeling of being wet. It's a neurological recipe.
Exactly.
Combine 1 part evaporative cooling from the TRPM8 channel, 2 parts tactile pressure, let them crosstalk in the S1, and your brain renders the reality of water.
And we can actually observe this mechanism dictate animal behavior. Consider a dog vigorously shaking itself off after a bath.
Oh, the wet dog shake.
Yeah, that classic wet dog shake is entirely dependent on this precise S1 integration. In laboratory settings, if researchers pharmacologically block the TRPM8 cool channel in an animal, preventing the brain from receiving the temperature half of the recipe,
The animal won't shake.
the animal will not perform the wet dog shake, even if they're physically soaking wet.
Because their brain isn't rendering the illusion. As far as their nervous system is concerned, they are experiencing pressure, but not wetness.
It's amazing. And we also see the devastating consequences of this integrated wiring in clinical neurology.
Oh, really? How so?
Well, patients suffering from multiple sclerosis experience a degradation of myelin, which is the protective coating around nerve fibers. This demyelination can cause aphaptic transmission.
Aphaptic transmission. What is that, essentially?
It's essentially a short-circuiting where signals cross wires.
Ah, so a touch signal might accidentally bleed over into a temperature pathway.
Exactly. As a result, MS patients frequently report vivid, spontaneous neurological illusions of feeling wet water dripping down their leg or arm, even when their skin is bone dry.
Oh, wow.
The brain is receiving the recipe through damaged wiring and flawlessly generating a physical reality out of thin air.
It really makes you wonder how many of our daily experiences are just mathematical synthesis. And temperature doesn't just mess with touch, right? It completely hacks how we process flavor, too.
It fundamentally alters the gustatory cortex. This phenomenon is known as thermal taste. Temperature actively modulates the amplitude of flavor signals.
How does that work?
Well, there is a specific ion channel on our tongue called TRPM5, which is responsible for detecting sweet, bitter, and umami flavors. Crucially, TRPM5 is highly heat sensitive.
Okay.
When this receptor is exposed to a warm temperature, its chemical structural conformation changes, drastically enhancing the electrical signal it sends to the brain regarding sweetness.
Wait. So if I eat a warm chocolate chip cookie straight out of the oven, it literally tastes sweeter to my brain than if I ate the exact same cookie an hour later at room temperature.
Yes, the objective chemical sugar content is identical, but the thermal energy amplifies the receptor's output.
Oh, that is mind-blowing.
And from an evolutionary perspective, this makes perfect sense. For our ancestors, warm food typically meant freshly killed prey or freshly cooked meals, items that are biologically safer, free of heavy bacterial loads, and incredibly calorie-dense.
So the brain rewards you with an enhanced flavor profile to encourage you to consume it immediately.
Exactly.
The brain is actively incentivizing survival through a sensory illusion. I even read that researchers can synthesize phantom flavors just by playing with temperature gradients.
They can. If you cool a subject's tongue down to a specific baseline and then rapidly apply a warm stimulus to the TRPM5 receptors, the sudden thermal gradient forces the receptor to fire.
And they taste something.
The subject will vividly taste sweetness, despite there being absolutely zero food, sugar, or chemical stimulus in their mouth.
You are tasting pure temperature. That is incredible.
Mm.
But, these cross-wired illusions aren't always pleasant or rewarding.
No, they aren't.
The Nature Review touches on the thermal grill illusion, which sounds like something out of a medieval dungeon, honestly, but it brilliantly exposes how the posterior insular cortex can just outright panic.
Oh, the thermal grill is a foundational experiment in sensory neuroscience. Picture a physical grill composed of alternating metal bars.
Okay, I'm picturing it.
One bar is set to a comfortably warm temperature, say 104 degrees Fahrenheit. The adjacent bar is comfortably cool, maybe 68 degrees. Warm, cool, warm, cool.
Right, so none of these bars are painfully hot and none are dangerously cold. They are completely innocuous.
But if you press your palm flat against all the bars simultaneously, you are hitting the gas and the brake at the exact same time.
Your peripheral nerves just bombard the PIC with wildly contradictory data.
Exactly. The TRPM8 cold channels are firing rapidly. The warm receptors are attempting to fire, but the disinhibitory gate is confused because the cold channels right next to them are fully active.
So the spatial summation in the brain breaks down.
Yes, the central nervous system essentially registers a massive systemic error. And to protect you from this unreadable, potentially dangerous thermal state, it recruits nociceptor, your pain pathways.
Oh, wow.
It produces a searing sensation of burning, noxious pain.
So you will genuinely swear your hand is resting on a burning hot stove and you'll pull it away even though the metal is totally safe to the touch. The brain defaults to pain is the safest illusion when the math doesn't compute.
It is an evolutionary failsafe.
This research obviously has massive implications beyond just understanding our own biology. Tech companies, specifically in the virtual reality and haptics sectors, must be devouring this Nature paper.
Oh, absolutely. The growing field of thermotactile displays, which include smart fabrics, advanced prosthetics, and VR gloves, is entirely reliant on mapping the posterior insular cortex.
Because if you want to create truly immersive virtual reality, you cannot just strap a vibration motor to someone's finger and expect it to feel like holding a steel sword.
Exactly, because we established with the doorknob that metal feels different due to thermal inertia and the rate of heat transfer.
Right.
To synthesize the reality of a metallic object, or the sensation of reaching your virtual hand into a stream of water, engineers must precisely trigger these nonlinear combinations of cool TRPM8 signals and tactile pressure.
They have to play the exact biological chords on the keyboard of your sensory receptors to force the PIC to render the physical sensation of metal or wetness.
The technological goal is to deliberately induce the hallucinations our brain already performs naturally. That is the ultimate frontier of human-computer interface.
Okay, let's distill this immense review down for the listener. What are the core takeaways from the invisible circuitry of our skin?
Well, first, cold is functionally dominant. It's faster, it fires sharper transient spikes, and it acts as the baseline alarm system deeply integrated with our sense of touch in the S1 cortex.
Right. Second, the perception of warmth is not an independent function. It relies on a disinhibitory gate. You literally cannot feel the warmth of the sun on your face without your nervous system simultaneously suppressing its cold sensors. The pathways are inextricably tangled.
And third, the physical world as you experience it. The wetness of a pool, the enhanced sweetness of hot food, the burning pain of a thermal grill is not a direct one-to-one recording of reality.
It is an active, brilliant synthesis constructed in the posterior insular cortex. Your brain is a movie director, taking raw thermal and tactile data and editing it together into a coherent physical narrative so you can survive.
We are never simply passive receivers of the environment. Our nervous system is a relentless generative engine.
Which leaves us with a truly provocative evolutionary question. If our TRPM8 cold channels and our disinhibitory gates are so precisely calibrated to the historical baseline temperatures of the Earth,
Yeah.
what happens as the planet warms? As climate change drives up global ambient temperatures over the next few millennia, our baseline cold brake pedal will be engaged less and less.
Wow, that's a fascinating point.
If our perception of warmth, wetness, and even flavor are all mathematically dependent on a cold baseline that is slowly disappearing, how will an evolutionary shift in ambient temperature fundamentally alter human perception of touch and reality in the deep future? Will water even feel the same to humans 10,000 years from now?
It fundamentally reframes how fragile and temporary our current sensory reality actually is.
It really does. Thank you for joining us in the Deep Dive Lab today. If you enjoyed uncovering the hidden algorithms of your own nervous system with us, be sure to follow and subscribe for more fascinating science stories. We'll see you next time.